有请唐纳德·英格伯教授。Welcome Donald E.Ingber.
Well, thank you for allowing me to join this esteemed group of speakers, which has been quite amazing. I have been a professor at Harvard University for 40 years. But for the last 15, I've been the founding director of the WE Institute for Biologically Inspired Engineering. It was formed in 2009 with the goal of basically inventing the future of bioengineering 30 years ahead. And when we started on this path, we looked back and we saw that engineering has absolutely transformed medicine over the last 50 years. Hip implants, pacemakers, drug delivery, you name it. But thinking 30 years ahead, we realized that while many people focus on how little we know, the fact is we've uncovered a huge amount about how nature builds, controls, and manufactures from the nanoscale up. And we realized that it was truly a tipping point, and that we could flip the paradigm. We can now leverage biological principles to develop new engineering innovations. We were kickstarted in 2009 with what was then the single largest philanthropic gift in Harvard's history of $125 million. And the donor, Hans-Jörg Wyss, challenged us to take on high-risk but potentially huge impact challenges. And the biggest problem that I could see at the time is that the drug development model is broken. Pharmaceutical companies spend over $250 billion a year around the world, and yet, to develop drugs, and yet over 70% of the time, when they get to human clinical trials, those drugs fail. In some areas, such as brain disease, it could be over 95% failure rates. So is there a better way? Is there a way we can develop drugs that are safer and do it faster and at a lower cost? And I think we can. And we're doing it with this little device. Now, what does this device do? What am I holding? Well, let me ask you, is it a device that can be implanted in animals to measure drug toxicity? Is it possible it's a new computer chip that uses AI to predict drug actions? Or is it a little device lined by living human cells from the organs of our body that can mimic the function of our organs for drug development? And so maybe to help you, let me give you a chance to take a look at it. So I'm going to pass this around. Who do I give it to? Let's see. I'm going to give it to you, but I want it back, okay? So let me ask the audience, who votes for A? Put your hands up. Okay. Who votes for B? Who votes for C? It's a very smart crowd, because you're right, it is in fact C. So what I was just holding up is what we call a human organ on a chip. Now, I'm not trying to build a whole organ in the lab. I like to think of these as living three-dimensional cross-sections or slices through a major functional unit of an organ. And to give you an idea how it works, let me describe our first chip, which we published 14 years ago, which was an organ chip model of a major functional unit of the lung, the air sac in our lungs. This is where you have gas exchange, where you have drug delivery, smog pollutants come into our body, pneumonia, COVID, lung metastases. This all happens in this part of our lung. It's a relatively simple structure. It's like a ball of grapes. It's hollow. It's filled with air when you breathe in. There's one layer of lining cells that are lung cells. Then there's a porous matrix that the cells adhere to. And on the other side, they're capillary blood vessel cells that make up your blood vessel. And this is where we exchange oxygen and carbon dioxide all the time. However, we also, every time we breathe in, they stretch. Every time we breathe out, they retract. And I've worked for 40 years on basic research showing that mechanical forces are as important as chemicals and genes in bioregulation. So at the WE, as I said, we try to use biological inspiration to engineer. So if we were trying to distill down this complex structure of a human organ, what are its minimal design principles? And we realized that a tissue are similar cells that come together to provide a particular function. An organ are two or more tissues that interface and new functions emerge. And almost always, one of the tissues is a blood vessel because you need oxygen and nutrients to survive. So if I can't show you what's happening in the device without looking through the microscope, but this video gives you an idea of how it works. The device, as you saw, size of a computer memory stick, it's made out of an optically clear polymer, or like a rubber-like polymer. If you cut it in cross-section, there are 3 hollow channels, each less than 1 millimeter wide. The middle one is cut into top and bottom by this thin, porous membrane, which we coat with matrix that cells stick to in the body. To make the lung air sac, we put human lung cells on top, human lung capillary cells on the bottom. We just recreated what we call the alveolar capillary interface. But the trick is we apply cyclic suction or vacuum in the side chambers. It's all flexible, and it stretches and relaxes at the same rate and degree as when we breathe. We could then flow air over the top, just like in the lung, and we could flow medium with or without immune cells through the bottom channel. And if we have cells on all 4 sides, we could even put blood for short times. Now, if this were to work, it should mimic an organ-level response. Imagine you have a bacterial infection, pneumonia. What happens is the cells on the lung side put out small molecules that stimulate the blood vessel cells to become sticky. And the white blood cells that were just flowing by now stick, migrate across, and engulf the bacteria. And that's how we keep healthy. So now I'm going to show you real images through the device. The white dots are fresh human white blood cells. I know they're fresh. We took them out of my postdoc. You can't see the capillary cells or the lung cells behind the screen because they're not labeled. To begin with, they're just flowing by. It's healthy. Now we add bacteria on the other side. You see that they stick. And it's important that the first sticking events are actually shear stress or flow dependent. So we wouldn't see this in a static condition. This is a higher magnification view. That's one white blood cell. It finds a space between 2 capillary cells. It goes through the pentagonal pore, and now it migrates to the lung air side. Now you're seeing coming out into the airspace. And now I'm going to show you the white blood cells in red because the bacteria are labeled in green, and you see them being engulfed. So you just watch the entire human inflammatory response in a lung-like context through this little rubber chip. Now, in our first paper, we actually showed that if you introduce nanoparticles that are similar to smog pollutants that we all worry about in air, that actually they induce, they're absorbed, and they induce inflammation and injury, but only when you have physiological breathing motions. If it were static, the way most labs study cells, you wouldn't see this. Pharmaceutical companies came to us and they said, this is interesting, but we're more interested in, could you measure drug toxicities, or even better, could you develop disease models, because animal disease models don't look anything like humans. So we hit 2 birds with 1 stone, as we say, by perfusing a drug called interleukin-2. It's an FDA-approved cancer drug. It's known as IL-2, through the blood vessel channel. And in patients, this induces fluid leakage into the airspace, causing a disease known as pulmonary edema. And when we do this in the chip, over a period of 2 to 4 days, you can see that there's fluid coming from the sides and filling the airspace over this exact same time period. Now, I mentioned that my lab has shown that mechanical forces are important for cells. And years ago, we actually identified an ion channel on the membrane of cells that mediates how cells sense and respond to physical forces. And we found a drug at the time that actually inhibited that response, and a drug company that was making it actually provided to us. And when we treated these lung chips, treated with IL-2, where you had fluid leakage, we completely prevented it. And in part, based on these results, that drug moved to clinical trials later on. But we didn't stop there. We then moved to a different functional unit in the lung airway. And this device mimics the ciliary motions, the mucus movement at the same rate as mucus in all of our lungs. And we could actually even expose it to cigarette smoke. We created a cigarette smoking robot. That's a cigarette lighter from a car and a Gatling gun type structure with real cigarettes. And the smoke, not extract, but smoke goes right into the airspace of the chip. And we've used this airway chip to model asthma, chronic obstructive pulmonary disease, cystic fibrosis, and the effects of smoking on our lungs. We've also made intestine chips. We've made models of the duodenum, the ileum, the jejunum, and the colon. And importantly, we could even grow complex gut microbiome on these chips for multiple days in culture. You can't do this in conventional cultures. If you put microbiome, it's called contamination, and the cells die immediately. But because we have this in vivo-like microenvironment where the cells spontaneously form intestinal villi, put out mucus, we can keep it in a homeostatic and balance with its gut microbiome. And this is very important because this is one of the major paradigm shifts in medicine in my career, that the microbes that live all over our body, inside and out, have a major determinants of health and disease. Now we can study it. The Gates Foundation saw the studies and asked us, could we model an environmental enteric dysfunction, EED, which is basically inflammatory bowel disease due to malnutrition seen in kids in low-resource nations. And we actually got cells from the intestines of these children as well as microbiome, and when we do that, we mimic the clinical manifestations of the disease, including inflammation, decreased barrier, integrity, even the gene expression profiles that we see in this little chip look like you see in patients. We've made a bone marrow chip where we take the blood-forming stem cells from the blood or marrow of a patient. We put it in a jello-like structure in one channel. We put capillary blood vessel cells in the other, and we feed it through the blood vessel channel just like our bone marrow. With this, we can get formation of white cells and red blood cells for over a month, very similar to what we see in our bodies. A different pharmaceutical company came to us because they had seen a very peculiar toxicity of a drug in phase 1 clinical trials, a cancer drug, that when they gave the drug over 2 hours, they saw low white blood cells and low red blood cells, but when they gave it over 2 days, they only saw low white blood cells. And they had a very hard time modeling this in animals. And they had measured how drug levels change over time in their patients in the clinical trial. This is called pharmacokinetics, PK. This is important because we all know doctors prescribe some drugs once a day, some once a week, some 3 times a day, and the frequency is very important because it really determines both safety and efficacy. So they had measured this, and you could see that in the graphs on the left side, on my left on the screen, in light blue. Because we have flow in these devices, when we perfuse the drugs through, we could precisely mimic the drug pharmacokinetics seen in patients, and when we did that, we precisely mimicked those regimen-specific toxicities. We then got cells from kids with a rare genetic disorder, and we're able to show that you can use these for personalized medicine effectively. These kids have a disease called Shwachman-Diamond syndrome, that they don't have normal red or white blood cell formation. When you put these cells on the chips, You can see that when you look at the whole length of the chips, there's just less cells. When you quantify, there are indeed less red blood cells, white blood cells, and stem cells. And we even discovered that there are two subpopulations of cells that have a slightly different defect that no one has ever seen before. Now, I'm just giving you a couple of examples because of time, but we and others at the Wyss Institute and around the world have made organ chips of virtually every organ that you can imagine. You can see a vaccination response in our lymph node chips. You can see metabolism in liver chips, excretion in kidney chips. You name it, we've been able to see it, and others are doing the same around the world. But when I first presented this idea over 10, 15 years ago, I predicted that we could someday build a human body on chips. Because we have a blood vessel channel lined by capillary blood vessel cells, you can imagine passing the liquid from one blood vessel chip to the blood vessel of another chip. And you can imagine giving, let's say, an oral drug into the intestine chip, watch it be absorbed, see it pass to the kidney chip and be excreted, move to the liver chip, be metabolized. Do you see toxicity in the bone marrow? And what does it do to a cancer in the lung? And we have actually done this. We've cultured over 8 different organs, liver, intestine, kidney, bone marrow, you name it. We had them all connected. We kept them alive for a month and showed that they were functionally connected. But we then used a simpler version of this human body on chips where we only had 3 organs. At the top, we have a gut, a liver, and a kidney, and then we added a little mixing chamber that is similar to blood mixing in your heart, so that if we take samples from it, it would be like taking a peripheral blood sample rather than sampling right after any one of the chips. And we used this to see if we could predict drug pharmacokinetics beforehand. This is important because when you go to clinical trials, you don't know what dose to use, so you have to gradually move up your dosing. It takes a very long time. If you could predict beforehand, you could greatly accelerate this process. What we're able to do is use computational modeling in combination with measurements of drug levels at every point in these different linked chips. And we could scale from the tiny volume and flow rates and mass of the chip to our body. And when we do that, we could show that when we added an oral drug, nicotine, like in chewing tobacco, into the intestinal lining of our intestine chip, we could quantitatively predict its levels, and that precisely matched what we saw what other people saw in patients in clinical studies. We did the same with an intravenous drug, cisplatin, a cancer drug, going through the blood vessel channel, and we precisely mimic what has been seen in patients. Now, exciting finding, and an important finding, is that about a year ago, we published a paper where we created 870 human liver chips that each chip had 4 different cell types. There was the lining cell of the liver, the hepatocyte, there was the liver blood vessel cell, and then there were cells called Kupffer cells and stellate cells, all in the right positions in this little chip with flow in both sides. These chips were made from 3 different humans. We tested 27 different drugs, most of which the pharmaceutical consortium had identified as being sort of the benchmark to say whether or not you can do better than animals, because they knew the results already, not only in animals, but in humans. And most of the drugs, the results from animals were wrong when they got to humans. And the results of that study showed that these chips were 7 to 8 times more effective at predicting the human response than animal studies. And in the same study, we did an economic analysis, and we estimated that the pharmaceutical industry would save between $2 to $3 billion a year by preventing late-stage failures just due to drug-induced liver injury. Now, these chips are important because they could also save costs. And the rising costs in medicine are killing us around the world. A biotech company that developed one of the COVID vaccines is now using these in-house to replace non-human primate studies, because non-human primates are, a major ethical concern, but also incredibly expensive, and now, since COVID, very hard to come by. And they showed that they can normally take 5 years at a cost of $5.2 million to test a number, I don't remember the number, but a number of drugs. And with the chips, they can do that in 18 months at less than 1/10 the cost, the same number of drugs. Now, it has important implications for the future. I mean, obviously, I've showed you already, we can make your lung on a chip, or your liver, or your kidney on a chip. We can make a human body on chips and see how the different systems interact. We can make disease models if we can get cells from patients that have particular diseases. We could do testing of drugs in cases where you can't do a clinical trial. For example, right now we do major research on radiation-induced toxicities. You can't do a clinical study with a radiation countermeasure drug because the radiation would be lethal. We could do that on chips. You could potentially do prenatal testing, very hard to do clinical trials on a pregnant woman. You could do old versus young. You could do female versus male. You could do children versus adults. You could also do different populations. But I think the real game changer for me is that right now, most pharmaceutical companies spend tens of millions, if not more, on clinical trials. They can do thousands of patients. They almost always fail. And then they'll go back and they'll look, was there some small genetic subgroup who responded better than the rest? If they find them, they'll do a small targeted clinical trial, and if they're lucky, they get approved for a very narrow application. Again, with organ chips, we could flip that paradigm. You can imagine that you take 50 to 100, let's say, patients from Sichuan who all have lung cancer. You make their lung chips with cancer cells in it. You make their liver, their kidney. You test drugs against those chips from those 50 to 100 patients to find, which subgroup of those patients respond the best to which drug and have the least toxicities in these other organs. And then you use those same patients for a small clinical trial. If that worked, we truly would be able to develop drugs faster, cheaper, and more effectively than ever in the past. Now, I'm very, very happy that I can share with you that, in part based on our work and others around the world who are doing similar types of studies, that the US Congress about a year ago passed the FDA Modernization Act, which now allows, for the first time since the late 1930s, the FDA, the Drug Regulatory Agency, to consider data from human organ chips in place of animal models. It's going to take a long time for the industry to take the chance to change the way they do things, but it's now possible. And finally, this may surprise you, but there's been interest in these organ chips far beyond pharmaceutical and biotechnology industries. The art and design world also are believers that design can really bring about better things for mankind. And the chips that I passed around are actually in the Museum of Modern Art in New York City, in their permanent collection, as well as in many museums around the world. So to end, I think my take-home message is that nature really has no separate departments of biology, chemistry, physics, or art, and that the real game changers, the way we're really going to solve the big problems, is by breaking through boundaries and taking on truly impossible challenges. And I hope that each of you will find your own path and join all of us who spoke to you today in this great tradition of science, which is really understanding how the world works to make it a better place for all. So thank you so much.